Well, it's great to be here, and I'm really excited to see all of these folks here. Every time I come, it's a bigger meeting, and that's a really great sign. I'm actually want to point out something to this group that I find is sometimes not well understood, which is that everything Anantha said is right about my background, but I'm actually a clinician at heart. As Anantha said, I trained in neurology at the MGH, and the thing that drove me back to the lab actually was very similar to what Petra talked about. Petra is a fellow neurologist, so as neurologists we are particularly privileged to see diseases that have no treatment and that are particularly horrific from my point of view for the patients who were afflicted by them. And that's actually what drove me back to the lab, because I had such cognitive dissonance that I could not imagine going through my entire career seeing patient after patient after patient for whom I could do nothing. And I had went on a mission to try to change that, and everything I've done since then has been in that because of that. Actually, that's why I did genetics, because like a lot of people back in the 80s, I was convinced that genetics would teach us a lot about the molecular basis of disease as a starting point for therapeutics. A lot of you know that I actually have worked in primary care, I've worked in community hospitals, I've worked in the third world, in community settings, and went to Merck because, especially at the time, I thought, well, you know, what environment is going to be forced to bring together the really elegant fundamental science with tremendous patient need? Well, it was a pharmaceutical company, because they have to create those interventions that improve human health, or they go out of business. And that was a really wonderful experience, and something that really informs everything I do today. But taking this job on about a year and a half ago really was the culmination for me personally of everything I've done in my career, because it brought me back to why I got into this business in the first place. And so though many of you may know me from the more recent work that I've done, realize that was done always with a vector back toward translation, back to the patient, and that's who's standing in front of you today. So what I'm going to try to do is to give you a very admittedly superficial overview of many of the things that NCATS is doing. It is a very ambitious organization. It's also a young organization. It's only about 2 years old. And so everything that I'm going to show you is really, think of it as a work in progress. But I'll try to give you a sense of how we're thinking about these problems as a sort of touchstone for where we're going. Okay, so as one of my mentors always told me, you know, whenever you give a talk, you always want to tell people what you're going to tell them, and then tell them what you told them. So here's what I'm going to tell you. So the first is, and you've heard this by multiple people already, translation is a team sport. But this is different from how at least I was trained. I was, and I'll go into this a little bit more in a minute, requires teams of individually expert members. But what strikes me and strikes many of us is that the current systems, particularly in academic medicine, often mitigate against this in a variety of reasons, and it's something that NCATS is committed to working on and changing. The second is that translational failure certainly is about 50% of the time scientific, that this process is remarkably empirical, from target validation all the way into implementation science. It's remarkably empirical. We do not understand the fundamental principles and the scientific principles which underlie virtually any part of the process, I would argue. And so when you don't understand the fundamental principles of a process, and you have a process which has so many degrees of freedom as this does, and you have a trial and error system, most of the time those trials will result in error or failure, not success. And so NCATS has got to establish the fundamental principles on which, the scientific principles on which translation is done. But even very clearly, if we get those scientific principles established, there are many non-strictly scientific issues which stand in the way. And there are incentive issues, there are collaboration issues, there are IP issues, a variety of things which also stand in the way. And so NCATS works on those as well. So novel collaborative structures, novel IP structures, public-private partnerships, these kinds of things. I'll try to get across to you that the opportunities, and for those of you who are younger in your career realize this, the opportunities are absolutely huge. Compared to when I was in training 30 years ago, the things that are possible now are much greater. And I know you're tired of hearing that, things that you could do when we all walk through the, all old folks, trudge through the snow to school. But it really is true. But the systems that we have for taking advantage of those opportunities are not there, in my view. And so we need to evolve the systems as well as the science. And they require systematic solutions. So what is NCATS? Where does NCATS fit into this? Well, NCATS is a very different beast by design in the NIH ecosystem. And there's many names I use for it, and these are just some of them. We're a catalyst, we're a convener, we're an adapter, if you will, from traditional fundamental research and patient-oriented or commercial research. We're an occupier of the tragedy of the commons. We work on problems that are everybody's problems in general, but nobody's problem specifically, so nobody has solved these problems, and I'll give you a list of them in a second. We're also NIH venture space. We have a modus operandi that says that translational failure is so universal that our default pathway is to do whatever the task is differently from how it's been done in the past. Why? Because the data are abundant that the way we're currently doing it is awful and fails. So why on earth would you do it the same way again and expect a different result? Which I think our psychiatrist, Anantha, would tell us is the definition of insanity. And we have a board-certified psychiatrist to tell us that. So the default pathway has to be, okay, what's a different way we can do this? And if you turn all your thinking on your head that way, then it really makes your brain activate a different way. So we have some core values. Because translation is a team sport, we have a core value of collaboration. Every project NCATS does is a collaboration with somebody. And secondly, we're really big on demonstrably useful deliverables. And by demonstrably useful, I don't mean demonstrably useful to the person who did the experiment only. That is, demonstrably useful because you got the paper, which got your promotion and then got you. That's great and essential, but demonstrably useful to somebody else. And that's somebody who is not necessarily in your field. And that's something that distinguishes translation, at least from the field that I trained in. I'm a developmental neurogeneticist by training, and I used to go to neuroscience meetings where there were 200 people in the whole world who did exactly what I did, and I hung out with them, and we all spoke the same language. That was wonderful, very insular. We all enjoyed it. That is not translation. And if you think about translation as a language concept, think of it that way. If you go to Russia, you have to learn what is interesting to the Russians and speak their language. That's very different and not how the typical ugly American acts. And I'll come back to that in a second. Okay, so what do we mean by translation? And actually a few years ago when I was trying to explain to Francis what translation was, when I got to NIH, I actually looked it up, and this is the definition, and I think about this a lot because it's useful. It actually means to carry across. So you're carrying something across to someone else. And of course, the problem with translation, and I want to pick up on something that Harry said in terms of advocacy, the problem with translation is it has many other terms, any other meanings to scientists, mRNA to protein translation, to my mother who understands translation is going from French to English. There are many other meanings of the term. So I go to Congress, and I don't know if you have this experience when you've been down there, but literally I have had congressmen and staffers say to me, okay, so I understand what the National Cancer Institute does because I know what cancer is. I know somebody in my family who has cancer. I understand what the National Eye Institute does. I don't really understand how the eye works, but I have an eyeball, and I have two. Most of them have two that I'm talking to. And so I understand that. But okay, so you're from the National Center for Advancing Translational Sciences. So what do you guys do, like linguistics? Literally, I have been asked that by a congressional staffer, linguistics. So we have a really important and different educational and advocacy process, because we have to explain to people our equivalent of what cancer is. So you think how difficult that is. We have to explain to people what the problem is and then tell them how we're solving it. So it's a multifaceted problem. Frankly, for somebody like me who likes big, hairy, difficult problems, this is right where I want to live. And this is if we can solve this problem, then our patients will take quantum leaps forward in health. But it is a daunting problem. Okay, so translation is different. So here's the ugly Americans in Paris. So this is what you don't want to do as a translation. But by definition, every successful translational project has importance to a stakeholder with distinct interests from the person who generated the result. So whether you're talking about a biologist and a chemist, or you're talking about a basic scientist and a clinician, or a clinical person to an implementation person, what they're interested in is different from what you're necessarily interested in. And they probably speak a different language than you do, and you need to think about what their needs are going forward. And this comes back to what a number of you know I talk about is how I think about community engagement. Community engagement at every level is absolutely critical to translation because of the nature of what translation is, to carry across. So again, this is very different from the insular nature of how most science is done. So what is biomedical translation? We've actually gone through a process at our place recently to actually define this. And believe it or not, we are in the process of using these definitions with various levels of people just within the institute, or within the center now. And we're actually using this with the assistants, the secretaries, to ask them, does this make sense to you? And then we're going, I've argued that we ought to have a mother round, so we'll send this to all of our mothers and say, do they understand it? And then we'll send it to any first graders who are around as a test drive for congressional outreach. So what is this? Well, translation is the process of turning an observation, an anecdotal observation in the laboratory or the clinic into an effective intervention which improves the health of individuals in the public. And this can be a diagnostic, a therapeutic, a device, a behavioral intervention. So what is translational science? So this is another important distinction. So every institute at NIH pretty much does translation. Translational science is what I was talking about before. It is the field of investigation that strives to understand the general principles which underlie the translational process, whether we're talking about how best to do medicinal chemistry, or how best to do toxicity or testing, or how best to recruit patients for clinical trials, or how to get patients to take their medicines. We don't understand what are the best ways to do this, and so therefore we fail most of the time. So NCATS studies translation as a scientific problem and an operational problem. Okay, so why are we doing this? Now, believe it or not, I am struck still by the fact that many people, including a lot of people in Congress, don't realize this, that we live in a very paradoxical time. Any of you who see patients, particularly patients with rare diseases, which are the ones that I've tended to see as a neurologist, but even the common diseases in neurology, think about it, schizophrenia, Alzheimer's disease, stroke, these are very poorly treated diseases. But we know more about ourselves in health and disease than we ever have, perhaps exemplified by the Genome Project, but our ability to translate that, to transform that understanding into tangible interventions which improve human health is really quite poor. And you see effects of this all around you. The drug-device diagnostic system is in crisis. The pharma industry, since I left Merck, has laid off about 250,000 people, about half of them scientists. And this is not because for lack of ideas or lack of opportunities. It's because this doesn't work as a business proposition because of the failure rate. I won't use the colorful language that Rob Califf uses to describe the clinical trials system in this country. I'm not even sure you'd call it a system. It's a clinical trials flea market or something. And so it's very clear, even if we had interventions for the 6,000 diseases that affect the human family that have no treatment, and that is the number, we would not have a system to be able to test these in any reasonable way. And even and Rob has really been one of the standard bearers here. Even when we discover interventions which improve human health in a hothouse environment of a clinical trial, we are terrible at getting that to every patient who needs them and therefore improving health, which is, after all, why we're doing this in the first place. And so what do you see as a result of this? Well, funders, people are unhealthier than they should be, first of all. If you want to be more depressed than you are when you read the Washington Post, you can read that IOM report that is pithily titled Shorter Lives, Poorer Health. It goes on for hundreds of pages about how unnecessarily unhealthy Americans are. But also funders of the process have either lost patients with us or are losing patients with us. And you see this in the private capital markets by the flight of capital from biotech into things like Facebook and other whatever Facebook is buying for $2 billion this week. But you see it actually in the public sector as well. And those of you who do advocacy know that the tenor of what NIH does, the attitude toward what NIH does in many quarters of Congress has changed rather dramatically in the last 10 years since I've been at NIH. And I think this is reversible. It's completely reversible. It's our game to lose. But I don't think, again, we can go on doing the same thing and expecting a different result in that form either. So I'm not going to spend a lot of time on this, but if you don't know this is another graph that just terrifies me. So this is Eroom's law. If you don't know this, it's an important paper for you to read, whether you work in this space or not. So you all know Moore's law, which is the improvement in the number of microprocessors you could fit on a transistor, which is responsible for why you could fit a mainframe into one of these now 20 years later. So this is Moore's law spelled backwards, Eroom's law, which is how the trend of improvement or lack thereof in drug development over the last 50 years, that is, that the number of drugs which is produced per $1 billion, inflation adjusted, has gone down by half in an absolutely monotonic way every 9 years since 1950. And this has been unaffected by the fact that development of computers, biotechnology, the birth of Rob Califf, all other there are no interventions which have affected this graph. And in case you're wondering, this graph goes to 0 in 2040, which means that there will be no drugs, or in other words, the cost of a new drug is infinity. So I would argue to you that much of this is a scientific problem, but this is a hard scientific problem. Now again, if you're interested in hard, big, hairy problems, this is the place to be, and here's another example of it. Okay, so into this maelstrom comes NCATS. Okay, so here's the mission. You've all seen this before, a catalyst to generate innovative methods and technologies to develop and implement improvements in human health. And I want to emphasize this catalyst issue. So NCATS is 2% of the NIH budget, so we have to be, like most catalysts, we're present in a very small amount, but we act by bringing together partners into proximity and allowing them to produce something which they otherwise wouldn't. That's what a catalyst does, a biochemical catalyst, and that's what NCATS does as well. Secondly, we focus on how to do each of these steps. And that's what, again, what a catalyst does. And we do individual projects. We need to do individual projects, but we're doing them as demonstration projects for these new ways of approaching these various problems. So this is the official mission statement that was given to me when I became director. I actually don't like it in a number of ways, but it will probably not surprise you to learn that to change a mission statement of an NIH center requires, it would take Harry, even Harry, it will probably take like 25 years to do this. And so we got, the people on the Hill, are, they're busy with other things like the ACA and stuff. So they don't want to deal with our mission statement. So, but we do have an informal but important modification that I've made in the red. So these, depending on whether how you do the definition, devices and behavioral interventions are not included in diagnostics and therapeutics, and they explicitly need to be. So I've changed this to interventions. And secondly, they can't just be interventions, which we say, oh, go try this, you'll get better. Because then in that case, we're no better than the GNC. I mean, they say that too, take the shark cartilage and you'll get better, right? So we actually have to improve, we have to show that these interventions improve human health, or our job is not done. And improve human health means people living, not people in a clinical trial, that's important, but people, living in their wild-type environment, in the sort of human equivalent of Wild Kingdom, where they're wandering around in their normal environments. A number of you have seen this slide before, but I always like to show it because it actually was made by somebody in Building 1, is the Institute Director's office, the NIH Director's office, as an organizational chart of the NIH. And it's absolutely true story. This is Francis's office made this, and I like to think of it as an aerial view of the silos that make up NIH. And you'll notice that NCATS is supposed to be a horse of a different color here, but it's also meant to be an adapter. So NCATS focuses on what is common to diseases, not what's different about diseases, what's common to the translational process, not what's different. So that is a completely orthogonal view to the rest of our colleagues at NIH. But because of the place in the ecosystem that we occupy, we also have much closer and more regular interactions with other parts of the ecosystem, disease advocacy groups, nonprofits, pharma, biotech, VC, FDA, et cetera. This is a slide that, or a diagram that I stole from the Harvard Catalyst website because I particularly like it. And I should say, I don't know if you can tell this, but from the back, but I am wearing my MGH tie today. So any of you from MGH, I don't know if Bill Crowley's here, but I wanted to make that point. And so NCATS, I don't need to explain this, but I usually do need to explain this to most crowds. So we work across the translational spectrum. Okay, so an illustration of how translation is a team sport. You've heard this. And for those of you who heard this analogy, spare me, or spare yourselves, go to sleep for the next 30 seconds as I go through this. So what is a team? A team is a group of people each of whom are top performers in a different discipline, and they work together to a goal which none of them can accomplish alone. So one of the things that really gets my goat is when I hear people in academia or pharma or anywhere else say, team science is not science because it devalues the importance of the individual. Give me a break. That means that Kobe Bryant is not a good basketball player because he plays on a team. The issue is that it's a different sport. So I would argue that fundamental science, and the way I got my training, because remember, I'm a geneticist, all my first papers were all two-author papers, you know, me and my lab head. This is what I grew up with. So somebody's got, Barry Kohler knows who that is. He's probably about the same age. Who? Sorry, Barry. Herb, you know who that is, right? That's Alexander Fleming, right? So it's got to be, right? You know, old guy, black and white, you know, old guy, you know, looking at crud on a petri dish, right? That's got to be Alexander Fleming, right? So this is the model that I grew up with, you know, lone investigator working away at night, you know, by candlelight in the lab or Bunsen burner or whatever, and aha, you know, you discover penicillin. And I had a number of those moments, actually, you know, 3 in the morning in the scope room looking at brains that I had worked on. And that's wonderful in the fundamental space, but I would argue that is science as golf. Right? It's a singular enterprise, perhaps with a caddy, otherwise known as a lab head. But that is not translation. Translation is football. And let me just take you through this because I think it particularly illustrates it. So in football, you have the two people who get the most attention. That's the quarterback and the running back, right? And the running back in this case is going to try to run around to the right side. And that number, see that white, the guy in the white uniform, I think he's number 13, he's in the defense. That's Mother Nature, who doesn't want her secrets revealed. And so this guy's going to try to score a touchdown, but he's going to get tackled by 13 because it's a very strong defense. All right. So number 72 there is an offensive lineman, and he's going to throw his body in front of the defensive lineman in order to allow his teammate to score, right? And it doesn't matter if you put 11 quarterbacks on the field or 11 running backs, you will never score, right? So now imagine that this was academia. So the senior author, who's the quarterback, the lab head, and the postdoc, who's the running back, if they score, the only people who are going to get credit are the first author and the last author, right? Number 72 is a middle author. Number 72 will not get tenure, right? So do you think number 72 the biostatistician, for instance, is going to go out of his way, throw his body in front of the defense in order to let his teammates score if he gets no credit? I don't think so. And then we wonder why we don't have translational touchdowns in academia. It makes perfect sense, right? And this system has got to change if we're ever going to make any headway. Okay, so what are some of the problems that we work on? And people sometimes ask me, well, you know, gosh, what are you talking about? What kind of problems here? If you look down this list, it is a relentlessly depressing list of general problems which bedevil the translational space, from target validation to implementation in the community. And you look at this list, and this is the sort of to-do list, partial to-do list that NCATS has. There's no disease name in any of these. And the reason is that every disease that you work on has all of these problems. So these are some of the problems that NCATS works on. And so one of the things that I've been beating into the NCATS staff, lovingly, but beating them into them, since I became director, is that it's important that we not only develop new approaches to each of these problems, but that we then demonstrate in individual use cases that they actually do what we say they do. Try them out, right? That's science, right? And if they do work, we cannot assume that whoever the community is, or who we want to adopt these, at any stage of the translational process, is all of a sudden going to stop doing what they're doing and adopt the right way to do it because we showed them. Right? So when people always ask me, what do you mean by that? I say, well, just imagine, you may have heard, if I show you in silhouette, I have been told that if I lose 15 pounds, that's good. Now, I know that's right. Somebody showed me. Have I done it? Of course not. Right? So the same thing applies. We have to have something called dissemination science to understand how to induce behavioral change to make people adapt these better ways of doing things. Okay, so I just want to. This is a bit of a non sequitur here, but I couldn't figure out where else to put this. There are a couple of things three things in our advisory council, just to show you kind of how I'm thinking about this. In our advisory council, we have I've chartered actually 3 different subcommittees to work on areas of translation that NCATS is, I think, are particularly essential to NCATS' mission. One of them is the area of patient engagement. We don't have time to talk too much about that today, but I think this is absolutely critical and a game changer in this space. So I have thrown down the gauntlet of our folks at NCATS to have systems by which we involve patients in every project we do from the very beginning. It is non-trivial to know exactly how to do that. Easy to say, difficult to do. So Margaret Anderson and Merle Weinberger, some of you know, are heading this advisory committee. NCATS has very little in medical technologies and medtech, diagnostics and therapeutic devices. And so Paul Yock and Frank Douglas are heading that. And then interactions with biotech pharma VC, Frida Lewis Halls, who's at Pfizer, and Ankit Mahadevian, who's at Atlas Ventures, are running that. Okay, so the programs and initiatives, and I'm not sure how much time I'm going to have to get through all this, but I'll just try to at least give you a sense of what we're doing. So the CTSA program you all know about, but then there's a number of programs in rare diseases. Why do we work in rare diseases? Well, rare diseases don't fit neatly into the silos that make up NIH because they tend to be multi-system diseases defined not on the basis of organ pathology, but on the basis of a genetic etiology, which tends to give multi-system manifestations. Secondly, I think about them as sort of the model organisms of the translational space, that they are simple systems, and if you're going to try to experiment on a system, it's better to do it in a simple system, and rare diseases are like that. And then we have a number of programs which are technology development programs to try to lift all boats, to develop, again, general principles of translation. Okay, so the Division of Clinical Innovation, and Petra hasn't seen this slide, so this is going to show you, this is the vision statement for the division that she's taking over. So Petra, pay attention. Okay, so it will not surprise you to learn, after the 3Ds thing I showed you, that the first goal is to develop, demonstrate, and disseminate methods and technologies that logarithmically improve the efficiency of the clinical translational process. Notice that word, logarithmic. I am really not interested terribly much in arithmetic improvements, a 10% improvement. And I'll steal something from Rob. who likes to remind us that many of the interventions in the cardiovascular space have taken, what, 15, 17 years to get to all the patients who need them. Okay, so 10% improvement, instead of 17 years, it takes 15 and a half years. Woo! Celebrate! Wrong. No, we got to go from 17 years to 1 year. That's a logarithmic improvement. And this is the case for all of translation, right? So when you think we got to have logarithmic change, a fold change, tenfold change, that again, like the do things differently as the default, it fundamentally changes the way your brain works. And so think about that. Emphasis on phenotyping. So as you know, I was originally a phenotyper, otherwise known as a clinical investigator and a clinician, and then went and did genetics. And you all know there's a lot of talk about genotype-phenotype correlation. And there's been a lot of effort in genotyping over the last 20 years, and that's been perfectly appropriate and incredibly successful. But now it is very clear that we can genotype with our eyes closed. I mean, I often tell Francis, his entire career, which is the Genome Project, you can now fit into a machine about the size of a toaster, because you can have toasters that do genome sequences now, right? So I often tell, Francis, your career has been boiled down to a toaster. But the problem is, in that ensuing time, given the love affair that the biomedical research enterprise has had with genetics, and I understand that, we have frequently lost track of the fact that the whole reason we're doing this is to correlate it with phenotype, and we have lost the expertise that I grew up with, and many of you grew up with, for a variety of reasons. And so it's very clear that unless we rekindle this and take it to the next level, we will never take advantage of the insights in genetics that have come. The training programs. I feel very strongly about this for a variety of reasons you might imagine, and having NCATS have innovative, and I really want to emphasize that word, innovative training programs that are different from what everybody else does, and thinking about where the puck's going to be in Wayne Gretzky terms. What are the skills, knowledge, attitudes that translational researchers are going to need? Development of an academic discipline of translational science. One of my dreams is, 10, 20 years from now, to have departments of translational science at universities that have the same cachet as the Department of Biochemistry or Genetics. Doesn't have that now, but to have those. And lastly, to really incorporate this idea of community engagement, communities across the translational spectrum. So you know about the CTSA program. I don't need to tell you about this, but this is the map now, and you notice that there is a new dot in New Hampshire. That's Dartmouth. For those of you who don't know where New Hampshire is, it's in the upper right corner. I don't know if Alan Green's here, but and I just want to show you something that's really, I want to thank a lot of you for, and something that has been really transformative for me in the last year and a half. This is a list of all the CTSAs I have visited since becoming director about a year and a half ago. I've visited 24 CTSAs and across the country, north, south, east, west, some new, some old, some big, some small. And there's 8 more. The ones in italics are ones that are upcoming, ones that I've agreed to do. And so here's actually the list. This is the red ones are the ones that I visited. The green ones are the ones that are upcoming. And the reason I show you this is that I really believe in what I told you at the beginning. Translation is a team sport. Everything is about collaboration. I have to understand what the issues are and what the issues that are important to those of you who are our partners in this grand mission that we are on. And so every one of the trips that I make to you have been tremendously useful to me, and I hope to make it to all of you before too long. So this is a report that you probably all read. This is a report from the IOM that came out last July. I just want to give you a little tiny update on this. So there were these 7 recommendations. You probably know what all of those were because it came out in July. But I just want to point this out. So One of the recommendations of the report was to work on how to implement some of these recommendations, particularly focused on the issue of measurable deliverables by which the program and individual CTSAs could be measured. And so we chartered a working group of our advisory council, which has a couple of members from our advisory council on it, Nora Desis and Scott Weir, but all the other members are not on the advisory council because we didn't have all the right expertise. But I don't know if you can see these names from the back, but it is an extraordinary list of people, from Gary Gibbons, who's the NHLBI director, to Fernando Reyes, who's a community engagement guy, to Bob Harrington, who's the chairman of medicine at Stanford, and a protege of Rob. Sorry, I keep picking on you, Rob. A protege of Rob Califf's. Really an extraordinary group. This group is completing its deliberations, and they're due to report to our council meeting on May 16th. And you'll get the report. We'll put it on our website, but you can tune in to the webcast and see this presentation if you'd like, but it'll be archived, and you can see the report. So I'm really looking forward to this as a next step in implementation of what the report says. So stay tuned on that one. Another thing we've done to instantiate this, what I believe is the second recommendation of the IOM report, that is to streamline the governance of the program. I've heard this over and over and over again from folks that having a steering committee, for instance, of 90 people, if you ever try to steer a bus with 90 people trying to drive it, it's very hard. So we last December, we created a new steering committee, and these are the members of the steering committee. It's chaired by the head of DCI, currently Elaine Collier, will be Petra Kaufman, and Nora Desis, who's the CTSA PI from University of Washington, and 12 PIs, altogether 12 PIs, so it's Nora and 11 other PIs, and then 4 people from the NIH currently on this. And the steering committee, I must say, is still getting its sea legs, I would say. It's a pretty new concept for the program, but this group, we don't have time to go into it in much detail now, they have been incredibly energetic and committed in coming up with particularly some really groundbreaking, game-changing consortial projects, which some, not necessarily all, of the CTSAs could contribute to induce this kind of systematic change, and you'll hear more about that as time goes on. And the other is that they've really been helpful in helping us think about the committee structure and how to rationalize that, how to maintain and improve the ability of individual folks at individual CTSAs and the NIH to work together, because of course, remember, translation is a team sport, core value of collaboration, without having this weight of required committees, which people told us over and told me personally over and over and over again was just taking them away from what they really wanted to do. So this is a work in progress. We're right in the middle of this, and I appreciate all of your patients who are involved in this, transition. Transitions are painful, but it'll be worth it. And I just want to show you that the steering committee has not been sitting on its hands. There's been, just since they were formed in December, there's been 2 in-person meetings and 4 conference calls, very active, and lots of email in between. So stay tuned on that one. So lastly, I do have to recognize Petra. What a joy to have Petra join us. And I just want to give her another round of applause. I realize a lot of you don't know Petra, but as you get to know her, you will know why I am so delighted to have her join us. She has got exactly the right scientific and clinical chops, and exactly the right heart, and exactly the right sense of mission that we need. And I know you will enjoy working with her enormously. She will be here, I think, for a lot of the day. Please do go and introduce yourself. She's anxious to hear from all of you, as I was and when I took over this job a year and a half ago. And you've been tremendously giving to me, so please do so for her as well. Okay, so just very quickly, what I'm going to try to do now is in the last 10 minutes, I'm going to try to take a supersonic view of the rest of NCATS. Because there's lots of things going on at the institute or the center that you probably don't know about. And so I'm going to run through a number of these very quickly. So first is that the preclinical part, I said every project there is a collaboration, and so this is the collaboration map of this part of the organization. And at 2:30 this afternoon, actually, will be a joint session between Barry Kohler and John McHugh, who's the acting scientific director of the preclinical division at NCATS, who will give you an illustration of how this actually works. I'd encourage you to go to that. I'm going to skip through a lot of this because we don't have time. This is where those staff come from. A lot of the expertise in the preclinical space comes from pharma and biotech, so that's where a lot of this expertise comes from. And you should think about these people as resources for you, because remember, they don't do projects unless they're collaborations. So I'm really anxious to increase the collaborations between the clinical piece and the preclinical piece. This part of the organization starts with target validation instantiated by genome-wide RNAi screening. This is a technology which was really not developed enough for routine use for a variety of reasons. This group was put together to solve the general problems of using RNAi in a genome-wide way to define all of the genes and pathways that are involved in a cellular process. And they've done that now, and all that work has been published, and they have a collaborative effort working on a number of diseases. This is a paper that was in Nature last December, looking at a bunch of new targets for Parkinson's disease, got a lot of press. The other thing that this group has done is to create the first public sector open database of genome-wide RNAi screening data. Believe it or not, now 10, 11 years after the Genome Project, there was no way to analyze genome-wide RNAi data because none of the companies who made the libraries agreed to make their libraries public. Without going into the details, I managed to, through some deft diplomacy that I think Harry would be proud of, I managed to convince the Life Technologies people that it was in their best interest to release all the data. And so this is now an enormous step forward so that investigators throughout the world can get access to these data. In the small molecule space is just one example, but there are many of these. This is an investigator at Florida International University who had a GPCR called relaxin receptors, involved in a number of things, including fibrosis, came to us, had no idea how to develop a small molecule probe or a drug. He knew a lot about relaxin, a lot about the biochemistry and the physiology of this physiological process, but nothing about the preclinical translational process. That's okay, because our people don't know how to spell relaxin, and they certainly don't know how to spell this investigator's last name. But together, they very rapidly developed the first agonist of this receptor, and we're very close to signing a three-way collaboration agreement to further develop this with a large pharmaceutical company. We do a lot of patient-driven science, and this is something which is, I know, of interest and importance to the CTSA program as well. We've done programs with the Charcot-Marie-Tooth people, with the Chordoma Foundation, and with the Myotonic Dystrophy Foundation to develop both targets and leads for their disorders. We put together a complete non-redundant list of every drug which has ever been approved for human use worldwide. I am very interested in using this resource to figure out what proportion of the 6,000 untreatable diseases might be treatable by something in the current pharmacopoeia. It's a really interesting question from a scientific point of view and a public health point of view. I'll just give you one example of how we've used this. It happens to be in CLL. What was done is to take patient cells from patients with CLL and normal B cells from the blood bank. This is what the data look like. Bottom line is that there were a couple of drugs, approved drugs, that killed all the CLL cells but didn't kill the normal B cells, which are the cognative CLL cells. One of these turned out to be an old rheumatoid arthritis drug called Oranafin. This is really important for a number of reasons. One is this drug works in CLL. It also has been repurposed to 2 other kinds of blood cancers since then, based on the common mechanism of this drug. But the other is that the reason this was never discovered before is that Oranafin is not a cancer drug. So the Cancer Institute actually has a group of approved, 800 approved cancer drugs. But this isn't in the collection. Why? Because it's not a cancer drug. And this is the point. The knee bone's connected to the leg bone, right? And if you look on biology, an integrated process, you can discover things that you otherwise wouldn't. So, and this has now been taken to the clinic in a novel collaboration with the Leukemia and Lymphoma Society and the folks who run the CTSA at University of Kansas. We've also developed new ways, and we'd be interested in talking to any of you about doing this, and these are the names I'll give you at the end, doing screening for novel drug combinations. A lot of you know, particularly in cancer and ID, but also in neuroscience, there's a lot of evidence that drug combinations are actually the way to go, but it's been very difficult to do this for technical reasons. So like the RNAi example and like the small molecule screening example, we've solved this problem. I don't have time to go into it. It was a combination of compound management and informatics, but we managed to do this, and here's an initial proof of concept paper that was published with Lou Stout, who's a cancer investigator on this kind of diffuse B-cell lymphoma. So again, a resource which all of you should know about. The TREND program is something that you might hear more about from John this afternoon. This is a program, again, a collaborative program that goes from lead to first-in-human studies. The previous studies that I showed you on relaxin, for instance, go from a target to a lead. This goes lead to first-in-human. But again, the same principle, collaborative principle. This is the scope. If you look at all of these things that I list here, a lot of them, some of you may not even know what they are, and that's kind of the point. Again, these folks don't know much about the diseases that they work on, but that's okay because they form joint project teams with the people who know about these diseases, who are disease experts, and the disease experts don't need to learn all of that they need to know in these spaces. But the important thing is that it is a joint project team that works on these projects together. This is the portfolio, and you'll have these slides. You can look at them. And I don't expect you to read all of this, but the reason I show you this is I want to give you a sense of the sociology. So if you look at the disease indications, they're tremendously broad, everything from sickle cell disease to cancers, to infectious disease, to neurologic myopathies, et cetera. About half of them are collaborations with academics, half of them are with small biotechs, and either repurposed drugs or new molecular entities. I'll just give you one example. This is on a particularly horrific pediatric neurodegenerative disease called Niemann-Pick C disease. It's a lysosomal storage disease, the gene for which was cloned about 15 years ago. And this was a repurposing of actually IT cyclodextrin, believe it or not, which is an excipient, never been used as a drug before, so very unconventional drug for a rare disease. But the only reason we were able to do this is if you look at the collaborators that did this over a very short period of time, this is what that project team looks like. This is 20 different people from about 15 different disciplines from 9 different organizations. And this organization, these people came together, they self-assembled because they were absolutely committed to developing a treatment for these kids in their lifetime, which is about 5 years. And we're in the process of doing it. So this can be done, but it requires a different modus operandi, is my point. If you're interested in more details about this, there's a really cool online feature that the Wall Street Journal did. There was a reporter that followed this project team for about 5 years. Amy Marcus, who's a Pulitzer Prize winning reporter, followed this project team and reported on it in a multi-part series that's freely available on the journal website if you want to see more about it. I'm going to skip this one because we don't have time. I just want to finish with a couple of things. One is on the Office of Rare Diseases. So this is another part of the NIH which was incorporated into NCATS when it was formed about 2 years ago, and its purpose was really the starting point for the NIDIS for rare disease work at NIH, sort of the equivalent of the Office of Orphan Products Development at the FDA, and the NIH Cognate was the Office of Rare Diseases. And it runs a rare disease clinical research network, which I really want you to learn more about and work more closely with now that Petra's here. And so these are its goals: facilitate clinical research. There's the Rare Disease Clinical Research Network. Facilitates clinical research, does a lot of both natural history and interventional studies. trains new investigators, much like the CTSA program does. But this is the structure of the program, and it's actually a very interesting concept of how this was put together. Each of these spokes on the wheel is a different consortium. Each of these consortia are multiple institutions which came together to not solve an individual disease, but rare diseases which have something in common, either a system in common or a biochemistry in common or a cell type in common. So they're not dealing with one disease, they're trying to learn general principles of this group of rare diseases. Secondly, you'll notice that at the top there's something called the Coalition of Patient Advocacy Groups. In order to be funded, you had to have patients and patient advocates as members of the research team, and this has been transformational to the efficiency of this program. I'm just going to give you one thing. So if you know Steve, Steve Groff started the program about 30 years ago. He just retired last month, but something that I want to make sure that you know, some people thought, well, Steve Groff's retiring, NCATS is going to lose interest in rare diseases. It's kind of bizarre, but anyway, that's what the sort of news out there. So that could not be further from the truth, that ORDR is going to continue its important work and actually go to the next level, even though its founders riding into the sunset. And if you don't know why Steve is riding a zebra, talk to one of your clinical colleagues and they will tell you. Lastly, a couple of things on toxicity. So toxicity is a common reason for drug development failure. So I just want to give you one really cool technology thing which NCATS does. So we're trying to understand how to predict technology. But this is really what I want you to tell you about. So this is a technology that or an approach that we hope will, as I was telling you before, logarithmically improve the efficiency of this step of the translational process. This step of the translational process is you develop a new intervention, a drug, a vaccine, whatever, and you put it into people, and you get unanticipated adverse effects that you don't understand. And so we're taking multiple approaches to that, but one of them is to make essentially a little homunculus in an in vitro system. That is to take multicellular organoid aggregates of either iPS, ES, or primary cells that represent important physiological functions of that organ, represent them in an in vitro microfluidic system, which can exist for up to a month in vitro, and then to connect those systems with artificial blood that allow you, for instance, to give a novel drug or a vaccine to, let's say, a novel drug, and you infuse this into this microfluidic system, and it goes to an artificial organoid, which represents the intestine and gets absorbed or not, and then goes to an artificial organoid of a liver and gets metabolized or not, then goes to the artificial kidney and causes damage or not, as an example. So this is a project which is a novel breakthrough technology, so it's classic NCATS, but it's also classic NCATS because it's a novel collaboration. It's a collaboration with DARPA, which is the Defense Advanced Research Projects Agency, and these are the organs that are being represented, and there are 10 organs, all the ones that you would expect targets of xenobiotic toxicity. And this is where the program is. It's a 5-year program. We're now through the UH2 part, and every one of these, there's 19 projects that are being developed representing different human organs, 19 different projects on 10 organs. And every one of these is a milestone-driven process, and every one of these is ahead of its milestones, showing you that the technology really is more robust than you might have thought, and I might have thought. We're now going into a UH3 stage here. For those of you who are NIH aficionados, you know what that means. If you don't worry about it. But the important thing is that in the first stage, we're working on individual organs. The second phase, we're working on tying these organs together into integrated systems. And I don't have time to go through this, but just to say that there's a lot that goes into this. Matrix, cells, perfusion in some cases, a variety of things. And I just want to show you one example of this. Right, so I love this program because it just, this has really, my mother can understand this. She sees the stuff going up and down. She says, wow, that's really cool. These are this is cardiac muscle tissue, and it turns out that if you represent cardiac muscle tissue and you just let it sit there in a plate, it actually doesn't develop the physiological processes that you'd expect because heart muscle likes to beat, right? Likes to move. And so if you require it to move, it actually develops the principles, the process of the characteristics of mature heart tissue, and you otherwise don't see that. It's very interesting. And we see this over and over and over again. And so eventually we're trying to create a body on a chip, and the cool thing about this is that you can imagine with IPS technology, you could represent not only a random person, but you could represent a chip for every person in this room. And do, if you really want to be out there, do a clinical trial on a chip, for instance. That's sort of the way that we're thinking. Okay, so, and I'm going to skip through this. I don't have time to talk about this. This is the Discovering New Therapeutics program. This is the collaboration with the 8 pharmas and the 58 compounds to repurpose these. There's a number of these that the CTSA program individuals or investigators are involved with. And the only thing I want to tell you about this is that there's a new round of this program that is going to go out on the street next month. So please keep your eye open for that. Okay, so take-home messages. I hope I've convinced you that the opportunities and needs in this space are huge, and they're systemic. And I really want to make sure that especially the younger people understand this, that it's needs, that's huge, enormous patient needs. But there are also opportunities. And so I think what it's up to, those of us who are gray heads or are old enough to have gray hair but have good genes, so I haven't turned gray yet, is to create an ecosystem where you can flourish to take advantage of these opportunities for the benefit of science and patients. That's really what NCATS needs to do. That the scale of these opportunities requires transformational, and these needs, requires transformational change that induce logarithmic improvements. And that we can't get to where we want, which is 20th century needs, can't be solved with 20th century solutions. And we really have to be creative on every level to make this happen, scientifically and operationally and organizationally. And lastly, and you'll hear me say this, everything at NCATS is an experiment. Everything, from the science we do to the way we're organized, so that we will continue to reinvent ourselves and to take advantage of these needs and opportunities. So we've just begun to transform ourselves and our programs to meet these opportunities and needs for the benefit of science and patients. So I thank you for having me here. I'm very excited about the next day and a half. And I look forward to continuing to partner with you to realize this very important mission we're all on together. So thank you.
Well, thank you, Chris. That was fabulous. In view of the scheduling and our time limitations, we'll probably not have any question and answer period with Dr. Austin, but he will be available and will be able to answer any of your specific questions that some of you may have. So thank you very much for attending this morning plenary session.